So, topic 6.3 transcription and RNA processing, some words that we'll get into. But first, I want you to take a look at this cell here and to see if there's some structures here that look familiar to you. Um, you should at least notice this big, huge thing in the center, the largest organelle in the cell. This is the nucleus, which contains our DNA. Um, if you'll notice, the next important thing we're going to talk about is something called a ribosome, which you'll find free floating in the cytoplasm or also in this rough endoplasmic reticulum out here. And we're going to actually be talking about through these next few videos, what do we use this DNA for that we find in the nucleus? Well, we use it to make proteins. That DNA is instruction for proteins. And this is actually known as the central dogma, right? The central dogma, meaning DNA gets transcribed, this process called transcription, into the single-stranded RNA, and this RNA gets translated into a protein ultimately. Reverse transcription is another process. We'll talk. I'll have you remember that this is something that can occur, and we'll talk about that in a later video. But for now, let's focus on the central dogma here. The central dogma. In other words, this is how life kind of generally works. DNA transcribed to RNA, translated to protein. Let's go ahead and review a little bit and look at some DNA and some RNA and look at some difference here. Look at some differences. You'll notice that DNA is double-stranded. Let me see if I can write that like double-stranded. RNA, single-stranded. You'll also notice a key difference over here. There's a different base in RNA than what you'll find in DNA, U and T, although they bond similarly. I'd like you to go ahead and, if you want to review yourself, are uracil and thymine pyrimidines or purines? Go ahead and pause and see if that review. Try and mentally remember which that is. If you said pyrimidines, you would be correct. You would be correct. Uracil and thymine are pyrimidines. Remember, those are single ring structures. But just keep this in mind. We're going from double-stranded DNA to single-stranded RNA in this first process that we're going to talk about. So first, I'd like to ask you just in general, and I want you guys to think about this. Maybe pause it before you move on too far. Pause it. Think about this. What is the word transcription? Have you heard that word before? What does that word mean to you? It doesn't have to be in the context of biology. Just what does that mean to transcribe something? To transcribe something. Okay, if you're back, maybe some of you did this and did what most people do. You use Google. This is what kind of the internet says transcription is: a written or printed representation of something. This first definition here, under here, this bullet point uses the word transcribing in it, but I think you get the point. The way I think of transcription is I think of a copy machine. Transcription is to copy something, to copy. So this is the process we're going to be talking about when we talk about transcription. How do we copy this DNA? We need to copy it and get it outside the nucleus to a ribosome where it can be translated. Well, we need to copy it into an mRNA form, single-stranded mRNA form. So let's talk a little bit about the different types of RNA that we can have. These other two on this side, I'd like you to recognize them because they're going to be important, but they're going to be important later on. This stuff, ribosomal RNA and transfer RNA, these are only involved in translation, which is a process we're going to talk about later. I don't want to waste too much time going over what those are yet. Plus, I don't want to overwhelm you. All we need to talk about right now is this nice little single-stranded stuff called mRNA or messenger RNA. mRNA, you're going to see that word a lot, so make sure you know what mRNA is. And it's called messenger RNA for a reason. This is what carries that copy or message out to the ribosome. Of course, it's a single strand of nucleotide bases with uracil instead of thymine. But this is an mRNA molecule, has this basic structure of being single-stranded like this. Okay, so let's look at this big complex picture right here, and let's break it down a little bit. What we're seeing here, you have something called the coding strand. You also have something called the template strand, also known as, and the curriculum wants you to know these other words for this type of strand, also known as the non-coding strand, the minus strand, or the antisense strand. So template, non-coding, minus, antisense, all of those mean the same thing. They mean the DNA that the mRNA reads to get that copy, right? Now you might be asking yourself, if the mRNA gets its copy from this strand, why is it called the non-coding strand? Isn't that kind of weird? Well, when you think about it, if this RNA here, which is going to be complementary, this RNA is going to be complementary to the DNA, right? That means that this RNA is going to look an awful lot like this coding strand as far as the basis goes. So that's why it's called the coding strand. This is what the code looks like on that mRNA. But the main thing to take away from this is that you have a somewhat similar process to what you had in DNA replication. The only difference here is you're not making DNA strands. You're unwinding DNA, right? And you're rewinding it at the other end. You're not making two copies. This process of rewinding really takes place when you're talking about DNA replication on those two separate strands. Here, we're basically opening it like a book, and we're copying some stuff from the middle here. Now, this molecule in purple here, this RNA polymerase, you should notice the word polymer and ACE. This is an enzyme that makes a polymer, or a long string of mononucleotides over here. So you have these triphosphate nucleotides down here. These are RNA nucleotides. And what's going to happen is this RNA polymerase is basically going to slam these onto a growing strand here, to a growing mRNA molecule right here. And this is what's going to give us our mRNA. Now, when it's done with this process, this transcription process, this DNA is going to, typically is going to rewind back up and become kind of normal, kind of standard DNA with the nice double helix like it might look over here. But what you should end up with is a long snake-like mRNA molecule. Now, of course, this is called pre-mRNA. And we're going to find out in the next video what pre-mRNA is. And how do we get from pre-mRNA to mature mRNA? But for now, I simply want you to understand this basic process of RNA polymerase attaching to a site, adding nucleotides to a growing mRNA strand out here, and you ending up with this nice long pre-mRNA molecule, which has a series of A's, U's, G's, and C's, right? And we're going to leave it at that for this video. And we'll come back in the next video and we'll talk about how we get from pre-mRNA to mature mRNA. Okay, thank you for listening.